To see how these genes are expressed in other cancers found in close proximity to the esophagus, we can simply add them back in using the tumor type filter. For example, let's add stomach cancers, or STAD. The heat map is instantly updated. We can see that many esophageal cancers cluster to the left with low levels of expression of these genes, while others cluster very nicely with other stomach cancers. Adding back the color bars to our sample set using change axis color bars, we see that esophageal squamous cancers are the ones that cluster on their own, while the blue, or the adenocarcinomas, cluster very well with stomach cancers. On the other hand, let's remove stomach cancers and look at the other end of the esophagus, closer to the head and neck. When we add this filter in, we see that many esophageal cancers cluster to the left, while others cluster very nicely with head and neck cancers. Adding back our sample set, we can see that the majority of samples that do not cluster with the head and neck are of the adenocarcinomas, whereas the ones that do cluster with the head and neck are the squamous cell carcinomas. Another way that we can look at expression data is the gene FPKM view. Under Select View, RNA-seq Quantification, Gene FPKM. In this case, gene FPKM will be shown in a box plot and trellised by gene, generating a single chart for each gene. These can easily be scrolled in the view by using the scroll bar on the right. Let's again look at these two cancers and also add back in stomach cancers. Notice in this view that stomach cancers generally have a large level of expression of any particular gene, while head and neck cancers have a much lower level of expression. Meanwhile, esophageal cancers appear to have a broader range of expression. We can use our sample set to split these cancers based by their subtype using multiple profile columns. Let's add our sample set to split the esophageal cancer group. By moving it over to listed columns, we now see that esophageal cancers are split by the grouping squamous versus adenocarcinoma. In this view, we can see that adenocarcinomas of the esophagus look very similar to stomach cancers, while squamous cell carcinomas look very similar to head and neck cancers. And we can scroll through multiple genes to see that this is generally the case. The examples in these videos have shown users how to easily filter samples of interest, generate custom sample sets to perform analytics within LAND, and finally, to display the results of these analytics.